And welcome to my presentation about the biodegradation potential of paracetamol by marine bacteria consortia. My name is Julia Valentine, and I was accompanied by my supervisors, Dr. Tanya Palma and Dr. Maria Costa, during this study. So today I'm going to be giving you guys a small introduction about my project, where I'll be including the methodology of the experiment, following with the results and discussions, and then I'll be ending with some concluding remarks taken from this study. So what exactly is paracetamol? Paracetamol, also known as acetamin, is one of the most widely used analgesic and antipyretic drugs all over the world. It was first synthesized in 1893, where it wasn't until 1959 that it was actually able to be used without a prescription. So some of the main physical and chemical properties of this pharmaceutical is that it is an odorless white crystalline solid that is actually soluble in water, about 14 milligrams per liter at 15 degrees. So in addition to that, its pH is around 6, so it does make it a slightly acidic characteristic. So some of the common uses of paracetamol is that it is used as an anti-inflammatory and a fever reducer. And during the COVID-19 pandemic, this is one of the most commonly prescribed drugs to help assist with the reduction of COVID symptoms. So for scientific purposes, I have pictured here the molecular structure on the right to represent the 4-hydroxyphenyl acetamin bonds. So how does paracetamol get into the environment? When paracetamol is ingested, the drug is not fully processed often, and it is excreted out of our body. So when it does this, wastewater treatment plants are actually not designed to remove the micropollutants from the wastewater, so they undergo an elimination process, which is limited due to the retention times that are actually very low within these wastewater treatment plants. So because these compounds are not fully able to be broken down, it leads to basically these pharmaceuticals having a direct pathway to the environment through the wastewater effluents. And the overall effect of this is not fully known, but it has been reported in many studies that they are becoming behavioral changes in the aquatic environment or different aquatic species. And there's actually trace amounts of these pharmaceuticals, such as paracetamol, being found in actual drinking water resources. So not only is that a problem, but paracetamol, when being degraded, produces intermediate metabolites called foraminethanol, hydroquinone, neutrophenol, and benzoquinone. So pictured here is a chart of the microbial degradation of paracetamol, which can produce foraminethanol, hydroquinone, acetate acid, formic acid, benzoquinone, and oxalic acid. So it's basically just showing you the pathway in which all of these are being created. And so all of these metabolites and these compounds are actually very toxic compared to their parent molecule, and they can cause a larger, greater effect in the environment if not properly observed. So you can see why this might be something we need to take into consideration. So what exactly can happen? So just as an example, foraminethanol is actually known to cause risks in damaging human liver and kidney functions. Hydroquinone actually impairs different cell function, which is essentially just weakening a human's immune system. So with the presence of these, it's obviously very toxic. Paracetamol and its metabolites have actually been known to affect the endocrine system of fish in the environment. So all of these things are cause for remediation. So what exactly is being done? Currently, aggressive chemical treatments methods are being implemented, such as advanced oxidation processes, to try and reduce the amount of these pharmaceuticals gaining access to our water resources. But this actually has a lot of negative side effects. So the alternative, which is something that we're going to be observing in this study, was that they are using microorganisms and particular bacteria that have demonstrated an important role in the biodegradation of organic compounds to reduce these pharmaceuticals being found. So this method is actually higher, it has a higher potential for bioremediation, it's significantly less expensive, and it reduces the amount of harmful chemicals being introduced to our environment compared to the AOP method. So that leads into my objectives. So the first aim of our study was that we wanted to obtain bacteria with the capacity to for environmental remediation. So using that, we wanted to observe analytical chromatographic techniques to help detect, separate, and monitor these concentrations of this pharmaceutical, paracetamol. And lastly, the aim was to grow bacteria cultures in the presence of drugs in order to select, identify, and identify the suitable bacteria for environmental remediation. So now looking at a little bit of the methodology, research collected samples from marine caves and mines, the mines being extreme environments. But for the purpose of this study, we only observed the 2 marine caves, the Cathedral Marine Cave, which 23 samples were collected from, and the Queijo Sucio Marine Cave, which 24 samples were collected from, both of which are found in the western part of Portugal, as you can see here in the picture. So the initial screening of the bacteria of the mine and the marine cave sites were done in order to check the biodegradation ability of paracetamol. The bacteria with the potential to bioremediate paracetamol was actually found, but like I said, only the samples, the bacteria enriched from the marine samples were used in the purpose of this study. Looking at the environmental or the extreme environments, those marine cave samples do need to be observed further, but here we have the 3 samples in which we use from the marine samples. We have sample 4, a porifera, samples 18, a tunicate, and samples 21, an annelid, all of which performed very successfully in previous works of the PROBIOMA study. So that is why in this part of the PROBIOMA study, we actually use these to continue with the study. So these samples were used to determine if paracetamol can be used as a sole carbon source, a carbon source being nutrients necessary for bacteria to grow. And so that's why we used these samples to conduct a biodegradation assay, where we subjected our bacterial samples to 2 different solutions, one of marine broth, which is full of lots of carbon sources, it's really rich in nutrients, and to MSM, which uses paracetamol as its sole carbon source. The negative controls were actually performed in the presence of the drug and in the absence of our inoculum, and this was actually to verify the interactions of the drug within the medium compound. So we also had positive controls, and these were made in the absence of our drug. So we did our study in duplicates, where we added the inoculum to 80 milligrams per liter of paracetamol in both the MSM and MB solutions. The cultures were incubated at room temperature between 23 and 27 degrees Fahrenheit or Celsius, sorry, and 150 RPM of orbital agitation, which was put in the dark so it could avoid all of the photodegradation. So from there, our 48 hours after incubation and our 168 hours after incubation was tested. So we determined the optical density which helps measure the bacterial growth present, and we also used the chromatographic techniques called an HPLC machine, which was used to detect and quantify the amount of paracetamol and its metabolites present. So once this part of the study was conducted, we actually wanted to determine the result of our objective 3, which was aiming to identify the suitable bacteria for environmental remediation. So we did that using biochemical and molecular techniques. So to identify these bacteria, these suitable bacteria, we completed a successive dilution protocol where we used 1 ml of our sample culture in 9 ml of 0.1% trypton water, and it was diluted among 6 tubes. So from there, we used 1 ml of each of those tubes, and we actually plated them on agar solid mediums using a method called the spread plate method. And so this agar was actually consisting of both the MSM and MB solutions, MB being our positive control, considering it has so much nutrients available. So here are some of the results that we determined during our biodegradation assay. The negative controls with approximately 86 milligrams per liter of paracetamol, which was detected from the HPLC machine, actually showed that there was paracetamol removal was actually very low. And so the mean of the condition was actually displayed here in the top left. The removal percentages for actually all of the bacterial consortium were significantly higher. And so then that means that the negative control, which basically it shows that there was removal of paracetamol and the bacteria was actually using the drug as a sole carbon source, using paracetamol as its sole carbon source. So during the removal, intermediate metabolites such as 4-methanol and hydroquinone were actually being produced. So pictured here is a chromatogram representation of the retention time of these metabolites after 48 hours. And then again, we have it after 168 hours. So you can see the peaks in which a furan, ethanol, and hydroquinone were present, and also the presence of paracetamol. So from the previous study, done at a lower percentage of paracetamol concentration, we actually repeated it using the two best samples, samples 4 and 21, and we increased the concentration of paracetamol, which was approximately 3,466 milligrams per liter of paracetamol as its sole carbon source. So we determined similar results, that paracetamol was able to be removed. There was the same metabolic products that were able to be detected, and in the negative control, paracetamol degradation was not detected. So moving on from there, we actually determined the bacterial isolates, which we observed their morphological types. So you can see the different coloration, sizes, and abundance of the bacteria within the plates. Many of these grew in the presence of the agar, inside of the agar, so we had to use special techniques to get these isolates out, which we then isolated them into their own agar plates to be grown. And then later we used DNA 16S RNA gene sequencing to help identify the exact bacterial isolates that were determined. So using the NCBI database, which is one of the databases that help us find the similarities between our bacteria and bacteria being found in these areas, isolates 1 and isolates 3 of samples 4 was determined as the genus Panabacillus, so it's a specific strand of this genus. And for consortium 21, we found similarities of 4, 6, and 7, or 8, sorry, that were comparable to the genus Panabacillus as well. For isolate 7, however, we had similarities with the genus Microbacterium, and all of these isolates have approximately 90 with our samples, which is really good. Just to note that samples 2 and 5 are under identification because unfortunately we did not have purity within our sequences, so we weren't able to determine any similarities. So that will need to be repeated. And so for some concluding remarks, it was determined that the bacterial consortium recovered from the marine organisms with the capacity to remove paracetamol was determined. And from those consortium, the main focus on the bacteria enriched from the marine samples, consortium 4 and 21 were able to remove approximately 80% of paracetamol, and it was using it as its sole carbon source after 168 hours. During this degradation of paracetamol, metabolites furan, ethanol, and hydroquinone were produced, so means for remediation are needed. And since the negative controls were paracetamol was in the presence of the inactive bacteria and in the medium, it didn't present actually a significant removal percentage compared to those in the presence of the bacteria consortium. So it should be concluded that the bacterial degradation, biodegradation seems to play the main role on the paracetamol removal. And lastly, we found 3 isolates from sample 4 and 5 isolates from sample 21 that were able to grow in the presence of 200 milligrams per liter of paracetamol, having similarities of around 98%. And so in the future, it is necessary to conduct a biodegradation study, basically using the bacterial isolates in order to understand which have better potential for this biological application. And so we would like to further observe the degradation of paracetamol and the products of its metabolites. So overall, thank you all for listening. I would like to thank supervisors Dr. Tanya Palma and Dr. Maria Costa for their guidance during my thesis. Thank you to the PROBIOMA project for your financial support during the study. Thank you to the University of Algarve and CCMR for assisting with sampling and providing us with a research team during our study. Thank you to the IMBRC Master's Program for this amazing opportunity. And lastly, thank you to all my friends and family for all of your support throughout this journey. So if anyone has any questions at this time, please feel free to ask.